Maas, Keezhapavoor.
Welcome to Maas Keezhapavoor channel. I am Dr. Aruna Shanmugavel. Let us see a motivational quote. Crying and trying have only one letter difference. Crying collapses our confidence and trying builds our confidence. So always keep on trying. The neuroglial cells are special connective tissue cells present only in the nervous system. They are non-excitable and multiply by mitotic cell division. The number of neuroglial cells are more than that of neurons, that is 10 to 50 times as much as that of neurons. The neuroglia are of two types, macroglia and microglia. The macroglia develop from neuroectoderm, whereas microglia develop from mesoderm. The macroglia present in the central nervous system are astrocytes, oligodendrocytes, and ependyma. The macroglia present in the peripheral nervous system are Schwann cells and satellite cells. Let us see the details of each cell one by one. The astrocytes are star-shaped cells with processes. These processes surround neurons and separate them from other neurons. The process ends in expansions in relation to blood vessels. The swellings present in the process of astrocytes are called gliosomes. The gliosomes are rich in mitochondria. There are two types of astrocytes. One is fibrous astrocytes and the other one is protoplasmic astrocytes. The process of fibrous astrocytes are thin and asymmetrical. They are seen in white matter. The protoplasmic astrocytes are seen in gray matter and their process are thicker and symmetrical. The astrocytes play an important role in maintaining a suitable metabolic environment for neurons. They are responsible for repair of damaged areas of nervous tissue by proliferation. This process is called as gliosis. The oligodendrocytes are rounded or pear-shaped bodies with few processes. Oligo means scanty. An oligodendrocyte may enclose several axons. The oligodendrocytes produce myelin sheaths to nerve fibers that are present in central nervous system. The ependymal cells line the ventricles of brain and also the central canal of the spinal cord. These cells are of three types. They are ependymocytes, choroid epithelial cells, tannocytes. The ependymocytes are the majority of ependymal cells. The choroidal epithelial cells are the specialized ependymal cells that are seen in choroid plexus. They secrete cerebrospinal fluid. The ependymal cells that line the floor of fourth ventricle have long basal process and these cells are termed as tannocytes. The microglia are smallest cells with flattened cell body and short process. They are more in grey matter than in white matter. These cells are closely related to capillaries and they are mesodermal in origin. The microglia act as macrophages to engulf and destroy the unwanted material. Most tumors of brain arise from neuroglial cells. Astrocytomas and oligodendromas are common. Right. The neuroglial cells in the peripheral nervous system are Schwann cells and satellite cells. Schwann cells are helpful in the formation of myelin sheath in the peripheral nerves. Remember, the same function is performed in central nervous system by oligodendrocytes. A well-developed Schwann cell is shaped like a rolled-up sheet of paper with layers of myelin between each coil. The inner layers of wrapping form the myelin sheath, whereas the outermost layer of nucleated cytoplasm forms the neurolemma. The satellite cells are small, flattened cells and present in the neurons of ganglia. They provide protection and nutrient support. They help to regulate the neuronal environment. Shall we end up this session with a quote? Performance always comes from the passion, not from the pressure. Always be passionate. Thank you for watching.
If you like this video, don't forget to subscribe and click the bell icon, select all to get instant notifications.